Good evening, everyone. Myself, Gaurav. Along with Sanjana and Tanmay, we have done our project on essential oil-based nanoemulsions against E. coli and Salmonella species in fruits and vegetables. Our project aim is to stop the growth of E. coli and Salmonella in fruits and vegetables using suitable and effective nanoemulsions of curcumin and tea tree oil. Novelty of our project is to combine the tea tree oil and curcumin and preparing nanoemulsions using volatile oils with the incorporation of nanoparticles such as curcumin. What are nanoemulsions? Nanoemulsions are colloidal dispersions of two immiscible liquids that have droplet size in the range of 20 to 500 nanometers. In this, oil acts as disperse phase, whereas water is in the continuous phase, which have increased stability and better penetration. There are also few non-pathogenic microorganisms, for example, Lactobacillus acidophilus, Staphylococcus epidermidis, Bifidobacterium linens, Bifidobacteria, and around 25% of the food in the world is spoiled due to microbial contamination.
This is the workflow of our project. For preparing curcumin nanoemulsion, we added 4 grams of curcumin, 3 ml of tea tree oil, 15 ml of almond oil, 8 ml of clean 1800 ml of distilled water. Then this mixture was sonicated using the following parameters. To prepare tea tree oil nanoemulsion without curcumin, we added 1.5 ml of tea tree oil, 7.5 ml of almond oil, 4 ml of clean 80, and 50 ml of distilled water. And then sonicated the mixture. Now this mixture was observed under a microscope at 40x. So for the antimicrobial test, we choose well diffusion method in which E. coli and Salmonella were spread plated on the Petri plates, and 3 wells were made for sterile water, nanoemulsion, and tea tree oil each. These plates were then incubated in hot air oven at 37 degrees Celsius, and the zone of inhibition was observed. We performed other tests like FTIR, particle analysis, and zeta potential on our emulsions. And these nanoemulsions were sprayed on fresh fruits to observe their morphological characters.
So these were the results that we obtained on our project. The figure 1 shows the nanoemulsion made with curcumin and tea tree oil, while figure 2 shows the nanoemulsion made just with tea tree oil due to the settling of particles in the first emulsion. This was the aggregation that was observed under the microscope, and that is not ideal for a nanoemulsion. So for the second, for our E. coli and Salmonella bacteria, we performed gram staining, and this is the view of E. coli and Salmonella at 100x under the microscope. And as for the zone of inhibition tests, which were obtained by well diffusion method, for E. coli and Salmonella, we can clearly see that the tea tree oil nanoemulsion has a large inhibition zone when compared to the positive control and tea tree oil. In this case, the positive control is water. So these are the functional groups that we obtained during our FTIR with the proper frequencies. This is the peaks that we obtained during the FTIR process. And as for the zeta potential test, we can find that the intensity is very high, which means that the surface aggregation is very low, which is ideal for an nanoemulsion. So these are the morphological changes that we observed in the following fruits. For apples, they remain fresh for 8 days, whereas bananas remain fresh for 6 days, and mosambis also remain fresh for 8 days. The following are the tables of interpretation of shelf lives of the fruits that we used. These all specify the fruits with curcumin, without curcumin, and control. From the following tables, we obtained a graph for the shelf life of the 3 fruits, where mosambis and apples stayed fresh for 8 days and banana was spoiled in 6 days. So the emulsion worked well for both mosambis and apples, but not as well for bananas. So this proves that our emulsion can work well on certain fruits, and this can increase their shelf life by a few days. Thank you.